Good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson. I'm the Executive Vice President here at Scripps Research. And today we have a really interesting topic. Reza Ghadiri will be telling us about the microbiome and how it affects our cardiovascular health. So Reza is a member of the chemistry department here at Scripps. Reza and I have known each other for a long time. I remember well meeting Reza. We were both assistant professors, the young Turks of the chemistry world, in about 1991 or 92. And I was at MIT, and Reza was at Scripps. And at that time, Scripps was not quite as well known as it is now. And he was one of the founding members of the chemistry department, which has now grown to be one of the top 10 departments. And I came to be here about 20 years ago, and so both of us remember those times fondly. So Reza is a chemist who applies his craft to biological problems. And so some of the work that he's going to talk about is modulating the microbiome within your gut using chemical approaches. Now, I think the microbiome has just sort of become this tremendously active research area. And I'm sure Reza is going to introduce a lot more about what that is. But in particular, the gut microbiome is something I think almost everybody that's listening must be acutely aware when they have had problems with their gut microbiome, and understand how important it is to get things back into proper balance. But I think what's only been recently appreciated is how integral the gut microbiome is to all of our other organ systems. And another example, we just hired Professor Howard Hang, who looks at the connection between the microbiome and your immune system. And he came across a pathway that links together those two areas, and he discovered it by studying the microbiome of cancer patients undergoing checkpoint immunotherapy, and was able to map this pathway. So it's difficult to underestimate the fundamental importance of the microbiome to human health, and in particular, it's something that we can actually modulate. And so Reza is going to tell us about some really fascinating work that he's done in this area, linking up the microbiome to the cardiac axis. So thanks, Reza. Please take it away.
Well, Jamie, thank you. Old friend, I'm really happy that you didn't show the picture of us sitting next to each other when we were young. Nobody might have recognized me with hair. So that's the, I'm pleased that I've been given this opportunity to present our work in this area. I'm going to try to just tell a story, more or less, why we did it and how it might impact some of the future research, rather than trying to bug you with lots of charts and data. First of all, I have to thank the people that have done this work. I'm a spokesperson for all of them. It is a highly collaborative research program across multiple departments here at Scripps, Janssen Research and Development, Salk Institute, etc., which I will acknowledge each contribution at the end again. So we all know heart disease is a major global problem. The rates of heart disease have decreased over the decades, but still is #1 killer in the US and in the world. Basically, every 30 seconds or so, somebody dies of a heart attack. Now, why are we talking about this? We all know that, if you have a low-fat diet, rich in fiber and vegetables and fruits, and if you exercise, you have a good chance of mitigating a lot of these heart disease problems. But We have lifestyles and habits that it's hard to overlook. Personally, I can't pass a juicy steak or a delicious burger, so it's very difficult to control these things. So there is really need for improved therapeutics in this area. But that's really wasn't the major objective when we started this. We were thinking of a microbiome, and you have seen this beautiful picture in the Scientific American. You know, we live with, let's say, largely bacteria and other microbes in and on our body. Actually, there are 90% of the cells that as we walking around, 90% of the cells in our body are microbial cells. So we are only 10% human walking around, believe it or not. And when you consider genetically and genetic diversity and the number of genes that are involved, we are about 1% human as we walk around. So you really should think about get out of the phobia of microbes. We really are symbiotes. Okay, we have co-evolved with the microbes in and on us. And I'm particularly going to talk about the gut microbiome, the bacteria especially that live in our GI tract. So there is no question that the gut microbiome, the microbiota, regulates our immune system, metabolism, and likely protects us against various chronic diseases. And by, another measure, if you consider somehow the microbiome is dysfunctioning, it has been correlated and proven that it associates with various diseases that are going through the entire human population, especially in the Western world, diabetes, obesity, heart disease, etc., including, which is really shocking, central nervous system disorders such as autism, Parkinson's, etc. So really we need to view a gut microbiome as our 12th organ system. Now it's easy to see why we missed it over many decades, because when you open up a human or another animal, you see all the organs, but you don't really see these diffuse microbes that live in our gut. But by any definition you can come up with, they act like an organ in our body. Okay, it's just not within a sac that you can ascribe it as in a pictorial way as an organ. So just like any organ that will dysfunction, will have consequences to our health, dysfunctioning of a gut microbiome, whatever that means. Okay, I'm going to use these terms very loosely here for descriptive purposes, but its dysfunction will have consequences. So there are several approaches to harness the therapeutic potential of a gut microbiome. Okay, I'm not going to talk about all the things fashionable things about diet and other things that people are recommending these days, because I'm not an expert in it, nor I find most of it scientific. But loosely, I can put efforts toward drugging the microbiome toward human health in 3 categories, roughly. We all heard about prebiotics. It's a nice word, which basically means fiber. Okay, so if you take fiber, which is largely complex oligosaccharides, they really promote growth of certain taxa and their metabolites, of which can be useful and so forth. So fiber therapy is generally useful. Then there are probiotics, which now multi-billion-dollar industry, and basically seeks to remodel your microbiome or add some bugs to it. The problem is potential safety issues, and more importantly, from scientific point of view, is engraftment issue. What does that mean? That basically the bugs, the capsule that you take that has few bacteria in it, they basically don't get a visa, okay, to join the community of your gut microbiome. So they get, after his resident time, it just gets washed out and so forth. So that issue, lots of great people are working on it, wonderful scientists, and eventually will be solved. So that way, then when you give a specific bacteria to someone, that bacteria can join the gut microbiome and augment its functionality. So far, it's a hit and miss, and it's difficult to do. The third approach falls in what we are all very good at, pharmaceutical companies, biotech, including us at Scripps, through Caliber, is drug discovery. Basically, it boils down to finding, identifying clinically relevant targets in the gut microbiome, or what the gut microbiome does to a target in the human side, and identify various gene expression, transcription, or proteins, enzyme receptors whatsoever, and try to drug it by coming up with a small or large molecule that interact with it and inhibits it or potentiates it, etc. So there's a lot of effort in that area, and all of these will eventually lead to some amazing new therapies. I would like to propose there's a fourth way of looking at this, and you have to be a bit brave and somewhat naive to even attempt something like this. And basically the idea is that since we know the dysfunctioning of gut microbiome, whatever that means, just I'm using these words right now for clarity. Since we know dysfunction of a gut microbiome can promote disease onset and progression, naturally the question is the opposite true? If I can remodel a gut microbiome from whatever this dysfunctioning state is, and I will explain what I mean by that, into something that is healthy, whatever that means, can you basically prevent the development of the disease? Is the opposite true? So how do you do that? Basically, everything that you put in your mouth is a selection pressure for the gut microbiome to remodel. If somebody's drinking 2 liters of Coca-Cola, lots of sugar, have pie for breakfast and lunch and dinner, naturally the gut microbiome will remodel to counteract or take advantage of the new type of nutrients that are coming in. In fact, many drugs that one takes will have an effect on the gut microbiome. So many things, supplements, this and that, can potentially remodel your gut microbiome. But all of those are not deliberate. You are not taking a gut microbiome from one state and deliberately going to another state. So the best example I can give you is, you get in a taxi, so the origin of the trip is known, and just tell the taxi driver, drive. You have no idea where you end up. If you do that the next day, it would be a different driver, and you say drive, you will end up somewhere else. So what I mean by directed remodeling gut microbiome is that you know your destination and where you're starting. So you instruct that I want to go to the airport. Okay, so that's a directed way. So all these other remodeling that takes place by simply, taking something or changing your diet, this and that, will shift your gut microbiome, but to what destination, I don't know. So we want to do it in a directed way. So the definition that I'm going to say is that any process, doesn't matter how you want to do it in general, that you can deliberately and in a predictive fashion change the state of your gut microbiome from one to something else that you have designated it. And you can do that by reprogramming its gene expression levels or by changing the composition of the gut microbiome. Having said that, I like to basically explain the approach that we're taking. So if we hypothetically, okay, basically there is, just a cartoon version. Let me see if I can have a laser pointer here. Let's say in this blue zone here, we call this a functional gut microbiome. And there will be lots of dots inside of this. Hopefully, most of you that are listening, your gut microbiome states, which depends on your bacterial compositions and the way their genes are turned on or off, as well as your genetic background and what you eat, etc. Hopefully, you will fall within, dots within this functional state. Now, on the opposite side of it, I call dysfunctional. So maybe I am over here in this place, and the gut microbiome is not functioning very well for whatever reason, and that would lead to some sort of a disease, such as heart disease in this case. The question that we ask is it possible to take a gut microbiome state that is inside this hypothetical dysfunctional state and somehow move it, change it, remodel it to fall within this blue zone that is a functional state? And in doing so, does it prevent the progression and onset of that disease? Okay, that's a big question, and there is lots of problems doing it. So first of all, if you're a scientist in the laboratory, you have to go beyond cartoons and you ask the question of what is functional, what is dysfunctional? It depends what you want to do. So we have to come up with a model for that. Fortunately, in the area of heart disease, there are really good mouse models that represent some of the things that happen in humans. In particular, the gold standard model is this mice that the low LDL receptor has been knocked out. Mice generally don't develop heart disease. But these particular mice, which has this receptor knocked out, they're perfectly healthy, happy, they have no sign of heart disease. If you give them the chow diet, which is basically a low-fat diet that most mice are fed, and they develop happy, healthy, and there is no sign of a disease. But if you put this animal on a Western diet, which is basically the average of what we eat, it has carbs, protein, fat, and a little bit of cholesterol. These animals, over the period of 6 to 10 weeks, develop high levels of plasma cholesterol, high levels of cholesterol in the blood, and develop plaques in their arteries. So most of the statins that people are taking, which has helped reduce cholesterol and incidence of heart disease in our population, they have gone through these kinds of models before going to clinic and studying it in humans. So this is a very good representation of heart disease in an animal model. And as I said, since whatever you put in your mouth, in this case, mice, will influence your gut microbiome, we expected that the gut microbiome of the mice that are genetically identical, okay, these are genetically identical animals, the gut microbiome of the animals should be different when it is on a low-fat diet versus on a high-fat Western diet, okay? Now, we call that a dysfunctional gut microbiome. It wasn't clear that the gut microbiome is responsible for the development of atherosclerosis. There is years of studies, longitudinal studies in humans, large population that show high cholesterol gets you heart disease and also inflammatory disease, inflammatory backgrounds, the inflammation and certain markers that go with it are responsible for development of atherosclerosis. So it wasn't clear that the gut microbiome, although we expected it to be different between the low-fat mice and the high-fat diet mouse, is actually responsible for this. But nevertheless, this will allow us to have a two-state model. We have the starting point, which is the gut microbiome of the animal that is in Western diet, that is fed Western diet, and the destination that we want to take these to, which is the gut microbiome of the animal that is on low-fat diet. So we have two states. for now, we call one dysfunctional, the other one functional. And in fact, when you look at the gut microbiome, you analyze from fecal samples or the content of the gut, intestine, and you sequence them, you get a picture like this. These are about 91 genera. In each bar here, in genus, there is several, in each, there's several bacterial species, okay? And you can see as you go, I normalized it, as you go from the chow and the difference into the high-fat diet, you see many of these genera go up and down. So within 2 weeks, the animal is on a Western diet, the gut microbiome is remodeled. So what do we want to do? What we want to do is take the gut microbiome of the animal that is on a Western diet and give something to the animal, okay, a drug, let's say, or a compound or a molecule that has the ability to remodel it into something that looks like it is on the low-fat diet, while the animal is still eating high-fat diet. Okay, so you got to be crazy to try something like this, but we are mad scientists, so we often do these kind of things. So how do you want to go about doing this? Okay, so what we came up is very simple in a sense. If you can have compounds that can selectively retard the growth of certain bacteria. Let's say when you're going from low-fat to the high-fat diet, certain genera increased in the population. So if you could selectively dampen the growth of those bacteria, then the others will naturally grow and provides a balance and moves the gut microbiome more toward the low-fat diet. The best analogy I can tell you is like horse racing. Okay, in horse racing, when the horse wins one race, the next race they put weights in its saddle to make it heavier. So the horse has to work harder, so in a way to normalize competitiveness between others. So that's what we want to do. We want to add a burden to certain bacteria selectively that overgrew in the Western diet, high-fat diet ones, such that the other ones have a chance of growing back up to normalize it more and look more like it was on the low-fat diet. Now, we have no idea how to decelerate. We have an idea how to decelerate, but we don't know how to accelerate the ones that got slower. So those horses will lose. Anyway, so here it is. We have a starting point. We have a destination. We want to know how to do this. How are you going to do this? Okay, you cannot use antibiotics. We all know antibiotics are broad spectrum activity. They just decimate the gut microbiome. And that's actually one of the problems of taking antibiotics. So how are we going to do this? And what is the, in scientific jargon, what is the mechanism-based rationale? I mean, you can't just take a shot in the dark. You have to have a way of doing this. So here is where I want to plug in the value of basic science. We all have heard that the basic science somehow enables something really interesting down the line. And we are all standing on the shoulders of giants before us, and science works this way. But in fact, here we have an example from my own laboratory. Around the time that Jamie was saying we were young, excuse me, we came up with this idea of self-assembling peptide nanotubes. These are cyclic peptides that are made up of right and left-handed amino acids. Natural proteins are made of left-handed amino acids only. So if you make it for various reasons, these cyclic peptides form these ring-shaped structures that, if the conditions are appropriate, they stack on top of each other to form nanotubes. So this was a basic idea we came up with for completely, totally different things we have never in my mind we dreamt of that these will be useful for remodeling the gut microbiome. I actually had no idea what the gut microbiome was 30 years ago. So what happens over these years, we figure out how to make them. These form beautiful tubular structures with these amino acid side chains, decorating it on the outside. And then we realized that if we can decorate the amino acids differently on the outside, you can have them go into membranes, such as lipid membrane, such as the membranes of living organism. And once it goes there, they can turn on their self-assembly program to sample these nanotube formation. And you can take advantage of it. Why? Because they can go into cells in many different ways, and they can either kill the cell or slow them down, because now they have an energetic burden associated with these kinds of interactions. Just like a horse, you're adding more weights to it, the bacteria, the bug will grow slower. The beauty of this is that each of these stacks that come together, one particular sequence will give rise to multiple codes for recognizing their environment. And in this picture, I basically have rotated one or more of these rings next to each other, just like ladies have two rings, and if you rotate one the other direction, the appearance completely changes. And basically it's like a jackpot. So they can present different functionalities to the cell surface to sample to see what they like. Is this the kind of cell that I like to interact with or not? And that's the really, without getting into too much details, is a source of how selective they can be. They don't behave like a general antibiotics will kill everything, almost everything. These things are selective for the cell types that they get into. So it is very easy to distinguish sequences that hit mammalian cells versus bacterial cells. But we found out, because we could basically pull the handle on this jackpot over the past couple of decades. We had lots of quarters to put in the jackpot. So we have rolled the dice or pulled the handle on the jackpot about 250,000 times. And during all those experiments, we realized that we can have a group of sequences that can care less about mammalian cells, but they love bacterial cells. And among those groups, there are some that like bacteria A better than B, or like C and D better than A and B, etc. So they can have a very sophisticated way of recognizing minute differences between bacteria, and they can selectively impact different kinds of bacteria. So we said that, is it possible that we can now apply these types of molecules to the problem of the gut microbiome that I mentioned? Can we find sequences that will selectively dampen the growth of these bacteria, for example, or these genera that overgrew in a high-fat diet, and thereby the other bacteria will grow naturally at their speed so that it will equalize it? Can we add a bunch of weights to many of these in the horse race, basically, to equalize the system? The question is, how do you find out what it works? Okay, you need to have something in the laboratory that you can screen for. What is the screening? So it took us about a year to develop this simple screen, which is you take the animal that has been conditioned for 2 weeks or more on a Western diet. Then you harvest the cecum, the content, the microbiome content of the animals, and you use it to inoculate every well within this 96 well. This 96 well is basically 96 test tubes, but very tiny test tubes. So they all have the same bacteria that came from the animal that was on a high-fat diet. As I said, that would be your origin. And then it took us about a year to figure out how we can grow this overnight and not basically lose all the diversity of the gut microbiome. So we can basically do this not too badly. And then what we can do then to add to each of these test tubes, little tiny test tubes, in this case 96 of them, different cyclic peptides that we had chosen based on our previous experience of how they modulate the growth of bacteria, sequence diversity, etc. And then let them grow overnight. And after 24 hours, we go in and find out what happened. Which one of these sequences dampened the growth of the bacteria or the genera that we wanted to slow down. How do we do this? We go and sequence each of these wells. Just like if you sequence your gut microbiome, my gut microbiome, and so forth. So we sequence each one of these. You generate an enormous data that then my bioinformatic colleagues will analyze and show that, for example, on the left, this is a peptide sequence, and on top you have various genes that are represented here, and you'll see on the side by how many log folds it went up or down, etc. And then you data mine this large data set using bioinformatics, cluster analysis, distance calculations, etc., to find out which of these sequences in the laboratory test tube turn this gut microbiome that came from a Western diet, turned it more toward the low-fat diet gut microbiome. And those will be then the candidates that we want to try in animals. To cut the story short, the animals, which are all genetically identical, they are born, raised to up to about age 10 weeks. And at that time, you put them on a Western diet for the following 10 weeks. Another group of animals will receive so-called placebo. The other group of animals will receive one of our compounds in their drinking water. So they can drink certain amounts a day. That's their dosage every day for the next 10 weeks. The beauty of these cyclic peptides is that they are abiotic in nature. They are made up of right and left-handed amino acid. So they cannot get digested, okay? They just stay in the gut. They cannot get absorbed in the rest of the body, just stays in the gut and comes out the other end when it's done. So, so these peptides are highly stable. So the animals will receive this are still on Western diet, okay? So you have, then you want to see what happens. So first of all, nothing happens to the diversity of the gut microbiome. When you analyze it 2 weeks later or 10 weeks later, it's not like giving them antibiotics, which will decimate the gut microbiome. There are plenty of bugs and they are very diverse in the gut microbiome. But very clearly, the state of the gut microbiome has shifted from the Western diet when you are giving animals also these peptides. And what happens, just don't worry about these data points. I'm just saying what happens in the animal is very similar to what we predicted in the laboratory tests. So the exciting thing is that within 2 weeks, the animals that are still on high-fat diet, their blood cholesterol, their plasma cholesterol drops by 50%. And all the bad lipoproteins, which you all have heard of, LDLs and VLDLs, plummet. And the good lipoproteins, which are HDL, which are basically the sponges of, in your blood system that remove excess cholesterol from your arteries, they increase threefold. So just like that, this is already better than, or as good as any statin that is on the market. But what's remarkable is when you look at the plaques in the arteries of these animals or plaques in the heart valves, etc., that can be analyzed in detail. Animals that are on Western diet have plenty of plaques. Animals that are on Western diet and our compounds basically are near the background, like if these animals were on the chow diet, low-fat diet. So there is close to 40 to 50% reduction in plaque buildup. This is exceeds the efficacy of some of the most statins that have gone through these animal models. So what has happened? Why, this is remarkable. How? What happened? Okay, for reasons that we totally don't understand, when you analyze the gene expression, meaning that what proteins and pathways the cells are turning on or off, we could annotate over about 60,000 genes, okay, in the gut microbiome, a third of which, okay, has gone up or down, meaning that there is a whole scale system-wide remodeling of the transcriptome, what genes are on or off. This new organ, which we call microbiome, has remodeled its gene expression. You can spend decades trying to unpack and understand this, but I'm gonna try to unpack one aspect of it. This remodeling of the gut microbiome impacts the host, mice. Hundreds of genes in the liver and intestine of the mice has gone up or down. We were interested, among many things, one thing in particular: what happened to the cholesterol? How did we get such a drastic lowering of a cholesterol in these animals? It relates to how cholesterol is metabolized. Now, cholesterol has got a bad name. Is an important molecule. Our cells need it. It is a starting point for various other molecules. It is an important, except the excess of it is bad, right? So our body, just like it metabolizes many other things, is also metabolizes cholesterol through various pathways in the liver, and the end product of it is what we all know is called bile acids. So there are dozens of different kinds of metabolized cholesterol into bile acids that are then secreted. The bile acids are, so here's the liver, bile acids are secreted into the intestine. And in general, one of the functions of bile acids is they act like detergents, okay? They act like soap to dissolve molecules that are not soluble in aqueous media, so that fatty looking things, molecules that have fatty surfaces can get dissolved and absorbed and some of the vitamins are that way and many things, okay? But that's not their entire function. This host of different bile acids, they are all what we call in science signaling molecules, meaning that they interact with various receptors and they are important in metabolism, homeostasis, this and that. There are many functions that these bile acids have. But one thing that happens is as the excess cholesterol or cholesterol is circulating in the body, goes to the liver and it gets metabolized into bile acid, and then other stuff happens and it excreted in the gallbladder and then into the intestine. 95% of the bile acids are reabsorbed back into the intestinal cell lining, and they end up back in the portal vein, which goes back again into the liver. Now, something very interesting happens. As you make bile acids get absorbed back and go back to liver. There is this complicated pathway that says, hey, we've made enough bile acids, okay? And it shuts down the metabolism of cholesterol, okay? That's normal feedback signaling for cholesterol metabolism. Made bile acids, then we detect bile acid in the liver again, and liver says, okay, that's enough bile acids. This is one of the reasons that high levels of cholesterol remains in the blood circulation because it's not being metabolized, because the signaling feedback said that let's shut down this metabolism. So here's something that was totally unexpected, but amazing that happened. It turns out, it was known that the gut microbiome that lives in your intestine also remodels and acts on the bile acids that are produced by the liver. So what happened here is because the gut microbiome was remodeled, that remodeling of the gut microbiome also remodeled the bile acid. And one of the members of the bile acid, beta-murocholic acid in particular, that particular bile acid is the one that turns off this pathway, saying that, no, wait a minute, don't shut down, keep metabolizing cholesterol, okay? So by the remodeled gut microbiome increased the level of beta-murocholic acid such that the metabolism of cholesterol remains on, and it keeps churning on bile acid, which eventually ends up in feces, how to get rid of the cholesterol. So that's how the cholesterol went down, which is totally related to the remodeling of gut microbiome. Gut microbiome also impacts the immune system through many pathways. As I said, many of the chronic diseases, including atherosclerosis, the inflammatory component, the immune response and inflammation is a major component in the disease progression. And remodeling of a gut microbiome through our collaboration with Janssen Pharmaceuticals, you clearly can detect various Tregs, etc., that are rebalanced. The immune, the inflammatory response is dampened. Many of the cytokines, pro-inflammatory cytokines, are now back to a normal level simply by remodeling the gut microbiome. I should remind you again, all these animals still eating their cheeseburger and fries. They're on Western diet, but their gut microbiome feels like it is not, okay? So therefore, everything looks like they're having more or less low-fat diet. So this little approach that I told you about, it shows, to me at least, definitively, that it is possible to purposefully remodel a gut microbiome and have an impact in a very complex and important disease. And in doing so, basically provides a proof of concept that there might be rational ways that we can go forward and provide new forms of therapeutics down the line to prevent heart disease and many other chronic diseases. So this was just an example in that context. It remains me to thank my co-workers and collaborators. You know, this work at Scripps would not have happened without my colleague, friend, and collaborator, partner in crime, Professor Luke Lehman, who is actually the one that suggested we dust off the cyclic peptides that we had worked on for unrelated projects and apply it to this project. The work started by a talented postdoc and my super talented co-worker, Audrey Black. And then, however, Benson Po Chen is the one that really did remarkable work bringing it to fruition. All of which have been supported in significant ways by my co-workers, Adam, Perba, Asad, and we wouldn't have worked on any of this if Bruce wasn't helping us to stay. Has over 4 decades of illustrious career in industry, so he tells us what's real or not. All of the bioinformatics stuff is done by my colleague, Ali Turkmani, and his co-workers, Bhuvan, for which we will not be able to do this multidisciplinary research without various metabolomic studies are performed by my colleague and old friend, Alan Sagatilian at Salk Institute and his colleagues, and all the immunology that I told you about, some of which I touched upon, was led by Dr. Pejman Surush at Janssen Research and Development and his colleagues there. But it wouldn't happen without the people that worked for about 30 years on self-assembling peptide nanotubes, the list of which is long. I think more than 30 super talented students and postdocs have worked on this. 11 of them are right now are professors themselves, conducting their own research and new discoveries, and collaborators at Scripps, my colleagues in other departments, Don Bashford, Duncan McCree, Ron Milligan, Glenn Nemero, and of course the funding from various sources. But before I close, I would like to say that you will have a really pre-Christmas gift. My colleague, Professor Valente, will knock your socks off talking about her most amazing work in the field of HIV, which I highly recommend for you to tune into that one, and it will be for sure very illuminating and exciting. Thank you again for having me here. It's the first experience for me to speak to a large audience without seeing you and talking to a computer. I hope things that I said made sense. And I'd be happy to answer questions relayed to me by Jamie. Thank you.
Terrific. Thanks a lot, Reza. During your talk, I actually had the opportunity to go back and find the very photo that you mentioned.
Oh my God. I didn't have hair then either.
that was going to be my comment.
Oh, yes.
This is a picture from about 1993 of the Searle Scholars annual meeting. A group of friends, Reza on the far right. This is me. I had more hair then too, but I had a lower basal level or a higher basal level of hair. And then this gentleman is none other than Jeff Kelly. All three of us were at different institutions at that time, and we all ended up at Scripps together. And it's just been a real blast. So there's a lot of questions I want to get to. I do want to say a number of you have requested sort of specific medical advice, and I really have to just sort of say that, look, this is a basic research talk, and we really are not in a position to offer medical advice based on this work, and you should really talk to your physician about that. But nevertheless, this idea has piqued a lot of people's imagination about the many connections there are. And maybe there's a few themes. I'm going to try to aggregate some questions for Reza. So one of the themes is, people are interested in how your microbiome, repopulates. And, you mentioned antibiotics. One of the questioners says, what happens when you do a colonoscopy where you drink a liter of fluid to sort of clear everything out? So maybe could you just say a little bit about what you know about the dynamics of how your microbiome repopulates after any kind of perturbation, and is it the same thing every time when it comes back?
Yeah, well, I should tell you, I'm not the expert in this, but I know enough to sound like I know what I'm talking about. Good. So first of all, when you're doing colonoscopy and so forth, and you're doing colon cleansing, it's largely you're cleansing the particular matter so that the scope works and you can see what's going on there. You're not wiping out your gut microbiome, the seeds of which are there, and they will be happily growing back. Hopefully not too difficult.
But the tissue, your whole gut is a very complicated tissue. Yes. And there's all these little niches and crevices and crypts. Yes. So that even antibiotics don't really wipe them out. They might kill most of it, but what comes back is based on the cells that are kind of attached to the gut lining, right?
Yeah, antibiotics is a little bit different. It is well known that after a course of antibiotics, you can get back to where you were before, but sometimes it's delayed and sometimes not, and you have now opportunistic infections that can take place because the gut microbiome also prevents, the same way that probiotics doesn't work, you know, pathogenic bacteria also have a harder time, like C. difficile and so forth, have a harder time integrating itself and finding a niche within a healthy microbiome.
Right.
But you give it a chance when you're decimating it with broad spectrum antibiotics. So those topics have been researched and studied quite exhaustively, very well by luminaries in the field. And there's a lot known about that. And, you know, I suggest that, you know, you read some of those things, not, you know, there's lots of things written about it, but
right.
it is, you have a way of getting your gut microbiome back if everything is okay. Okay.
Right. So here's a kind of a specific question. So the experiments that you described is that that you changed the diet of these mice and then looked at what the microbiome did. So is there a way to put a specific microbiome under a constant diet, maybe the high-fat diet, and see what different microbes do in response? So sort of turn the experiment around.
Well, that's in a way, I'm not sure I understood the question, but that's in a way we did this, because when you condition the animal to Western diet, okay, and then analyze the gut microbiome, that's you know, the source. That's where you.
I think the idea was to challenge the gut with a different set of microbiome. So maybe like a fecal transplant.
Yeah. Yes, exactly, and those have been done in other contexts. Like you take these germ-free animals that are born in the bubble, basically, and there is no gut microbiome, and that's one of the you know, types of important experiments in this field, that then you can implant different gut microbiome in these animals and study what happens, right? The problem with that is that, you know, you should, I didn't try to belabor this point too much. Listen, you know, gut microbiome is so important in training the immune system and development of the immune system. The studies have shown that if a child born up to age 3, roughly, if he or she doesn't have a functional gut microbiome, he or she is likely will not develop a competent immune system the rest of his or her life. You know, so so these germ-free animals that are born germ-free and so forth, they have lots of problems. Okay, their immune system is compromised, this and that. So my personal feeling is that those kind of studies are super important in the right context. But in the context that we were trying to look at, we wanted to be as realistic as possible. I mean, nobody in the general population is germ-free. And most people have developed competent immune systems. So we wanted to start with animals that are not only genetically identical, but they have been, you know, raised and are normal to begin with. And then ask this question, can we turn around a diet-induced development of atherosclerosis? And we showed for the first time that it's really the a big component of it is the gut microbiome. It's not just the cholesterol. Okay, so that was the aim. But those can be done, and people have done those kind of studies.
So let me ask a really forward-looking practical question. So you described this really, you know, brilliant series of basic research experiments that just show clearly this connection between diet and the microbiome and the effect of these peptide nanotubes on the blood cholesterol levels. So everyone's wondering, well, you know, how do you turn this into a drug? So I know you've at least thought about that, and maybe it's a ways off, but maybe you could just describe what would be the path from where you are now to having something on a shelf where people are eating peptide nanotubes to fix various maladies.
Well, as we all know, there is a big chasm between proof of principle and clinical outcome, and really talented people have to navigate that chasm. But fundamentally, what I see this is there shouldn't be any impediment. Now that you know this can be done, perhaps you can screen for your favorite molecule. It doesn't have to be our cyclic peptide nanotubes. You can do your own discovery using the tools that I suggested, and I actually improve on the tools that I have suggested and we have used. But as far as our own peptides are concerned, it can be done if people choose to push this forward and is interesting to the pharma and biopharma. We just need to increase the activity of them tenfold, 10 times, because that way you're basically taking a 500 milligram pill a day. And right now you don't want to take 3 horse pills a day. It not just cost effectiveness, but compliance, but it can be done. Okay. I would say the caveat is, we chose a heart disease, not only we were familiar how to work in this area of atherosclerosis, but we chose it because it is such an important and difficult problem. But actually from the drug development perspective, it's the worst choice you can have because the clinical trials are very expensive. So not too many people want to spend $700-800 million and 4 years and large studies to come up with a new heart disease pill, which we needed, but so there needs to be some people that are brave and with deep pockets that want to do this.
One of the other questions that came up in a couple of different ways is, there must be an interplay. There's a third axis, which is your genetics, you as a person. So maybe could you talk a little bit about genetics, the microbiome, and your ability to modulate?
Exactly. So if I want to be forward-looking and to the future and so forth, really what you want at the end of the day is a personalized medicine that you look up the list, compare your genetic background and other factors and your gut microbiome, this and that, and analyze and say you are on a path toward this chronic disease, okay, where you need to be over here instead, and have a personalized medicine. So it would be, so let's say you have a dozen of these as an example. This is totally hypothetical. You have a dozen of these cyclic peptides that remodel the gut microbiome different from each other, etc. And then you map one to the person based on those data points, and you say, okay, you need to take this for the next following 2 years, okay? And to see, because I said this, we are symbiotes, but even forgetting about that, this is another organ in your body. And we all have a little bit different genetic makeup, right? So if you think about that, then everybody's a little bit different from each other, okay? And how different there are lots of great people working on these things, but eventually we should be able to map people in different groups, different states of the gut microbiome, and how that relates to their progression of disease, and then have personalized medicine for them, okay? And that's like a big dream, but really we're gonna have this in the next 20-30 years. It's the future of how therapies should work, okay? Not one pill fits all, okay? It should be directed to different populations. That's how I see it. Will be beyond my lifetime, obviously, but that's where it's going to go.
So there's another theme that I could pack it all into one kind of question. And so people are interested in other systems that might be equally as impacted, other organs, liver, heart disease, what about aging? Everyone basically is really intrigued by this relationship. And so So, obviously, this is your particular focus, but you read the literature. Maybe you could just summarize where this is all heading.
I don't know how to deal with the aging part, but it's a matter of quality of life. I mean, I think what science and technology and engineering and other levels of and areas of science have provided to humankind is better, hopefully, quality of life. Okay, and the fact that we are zooming right now, it's the marvel of engineering and programming and years of scientific research that has gone to this, so we can communicate better. So I really think that these sort of things will work itself out. Okay, so and how impactful this can be is, I would say that you are seeing the impact of it to a large extent because it was a neglected area. Okay, we really didn't have the tools till the advent of next gen sequencing, so that we can sequence faster, cheaper, better. We really couldn't do the things that I told you today. Okay, it would have been prohibitively expensive and time consuming. It would have taken 10 years for me to, I wouldn't have been able to do it in 10 years, analyzing the gene transcription and the microbiome in those 96 wells. Okay, so it, for various reasons, okay, it has been a neglected area. Therefore, now when you're starting to dig in it, you will see many significant avenues that you can pursue for human health. And it's just natural that there is so much interest in it. Unfortunately, when there is interest, there are so many brilliant people working in this area, excluding me. No, seriously, there are so many wonderful people working in this area that's going to make an impact, okay, one way or the other. You just, to the interested audience today, you just have to separate the fashion and advertisement from what's real or not. And that's often difficult, okay? So a lot of things that are now piggybacked on the microbiome may not be useful and could potentially, I don't know, could potentially be harmful actually. So be careful about this. So I suggest in the meanwhile, eat,
I mean,
as well as you can, and exercise. Okay.
A safe way to say it is that it's very complicated. There are many different factors, your genetics, your diet, your age, your health level, that intersect, and so there's no one size fits all. But there's an overwhelming number of questions between Q&A and have. There's over 100 questions, but we're going to have to wrap it up here. And so I'll sort of end on the last one, and it's in a way, a couple of people have asked this: how can I figure out what my microbiome is? So what's the home microbiome test?
Well, I don't know. There were some companies that you could send your fecal sample for gut microbiome. A couple of them don't exist anymore. Maybe there are some new ones out there. I really have
basically it would have to be sequencing, right? Right now, there's the only way to do it is collect the cells and send them off for sequencing to identify
the question is, what are you going to do with the data?
And now what do you do?
Yeah, right. But there are companies that are trying to correlate those data to various other health status and markers and your medical history, etc., to make some sense out of it and make some suggestions. I think it's a good start. I don't know where it's going to go. Okay.
We're way off from the kit you pick up in the supermarket to, we're way off, right. Well, Reza, thanks a lot. There's been an overwhelming interest in your talk. You gave a brilliant lecture. And if I might personally reflect on 30 years ago, neither of us could possibly have predicted what we're working on now when we first met 30 years ago. So it's been a wild ride. So thank you all for joining. Join us for Susanna Valenti at the next front row. And in the meantime, be well. Goodbye, everyone.